අපි අපේ බෙච් එකේ ට්‍රිප් එකක් යන්න කොහේ හරි වන් ඩේ නැත්නම් හාෆ් ඩේ.
Suja, good idea. Nimi miss ඉන්නවද?
වහිනකොට ට්‍රිප් යන්නේ මොකද? නිසරේ
Yes.
මේ සුජාට වහිනකොට ට්‍රිප් යන්න හදක් කරදී. මොකද?
නෑ නෑ
මේ
ඊළඟ අවුරුද්ද
මේ
අවුරුද්ද යන්න දැන්
අවුවේ
plan කරලා දාන්න.
ගියොත්
සිංහලෙත්
නේ
බෑ,
අපේ
සිංහලෙත් බෑ.
beautiful ladies ලාට problem skin care එකට වහිනකොට ගියොත් no problem.
no problem.
Dr.
ඒකයි.
Nistri. Dr. Nistri.
ඔව් නිමි මිස් කියන්න.
ඔයාව host කරලා තියෙන්නේ.
ඔව් මැඩම් ආවහම මැඩම
ඔව්.
co-host කරොත් හරිනේ.
host කරන්න recording දාන්න.
හරි හරි. okay okay.
ඔයාට ඔක්කොම
දැන් මම
කරන්න.
recording නැද්ද? දැන් recording නැද්ද?
දැන් recording දාලා නැහැ. recording එක දැන්පත් තියෙනවා නේ මේවත් record
ඔව්
වෙනවා.
තියෙනවා. ඔව් ඒකයි. හරි. මේ සුජා කතා කරපු ඔක්කොම රෙකෝඩ් වෙලා පරිස්සමෙන්.
ඒක මේ මම record එක මේවා කරන්නද?
ඒ ටික ඩීල් කරන්න අලුතින් පටන් ගන්න.
Dr. Nistri ඔයාට තමයි මම ඔයාව host කරලානේ තියෙන්නේ. කරන්න පුළුවන් දැන් ඈ.
හරි නිමි මිස්. thank you නිමි මිස්.
එහෙනම් මම log out වෙනවා.
මොන හරි ප්‍රශ්නයක් තිබ්බොත් කතා කරනවා. ආය මේක මේ 40 minutes එකෙන් log off වෙනවද 40 minutes
එහෙම
වල?
මුකුත් වෙන්නේ නැහැ. එහෙම මුකුත් වෙන්නේ නැහැ.
හරි. okay thank you miss.
මොබයිල් එකට ගන්න මේ අද conversation batch එකේ.
හරි මොන හරි තිබ්බොත් විතරයි නැත්නම් no okay.
නිම්මි මිස්ටර් ඇබ්සන්ට් දානවා.
එහෙමද? හරි.
මට ප්‍රසන්න දාන්න නෙමෙයි මිස් මට ප්‍රසන්න දාන්න.
නිම්මි මිස්
batch එකේ
නිම්මි
conversation.
ඇරලා තියනවා.
නිම්මි මිස්
පේන්නේ
එලා නිම්මි මිස්
Okay.
මගේ මේ මම හිතන්නේ iPhone කියලා වෙන්නේ නැති වැටිලා ඇත්තේ නම තිබුණ නැහැ චුට්ටක් එක බලන්න
චුට්ටක් බලන්න කියන්නේ ඉතින් iPhone එකේ නම rename කරන්න නමින්.
දැන් කරන්නම් ඉස්සෙල්ලා ඒක උදේ ඒක
හරි.
ඒක පොඩ්ඩක් බලන්න ගිය සැරේත් ඒක වුණා චුට්ටක් ඒක බලලා දෙන්න ගිය සැරේ ඒකටත් iPhone කියලා වැටලා තිබුණා
හරි.
මගේ නමත් iPhone කියලා තමයි වැටෙන්නේ.
හරි.
මේ බලන්නකො iPhone තියෙනවයි කියලා කට්ටිය කියනවා. අපි නම් දුප්පත් මිනිස්සුන් අපි ගාව තියෙන Samsung phone එක. හරි.
Dr. Nistri මම log out වෙනවා එහෙනම්.
හරි මිස්.
Hello.
Yes, ma'am.
May I start?
Ma'am, hold on, ma'am. I'll make you as the co-host so you can
Yes,
share.
yes.
Yes,
Thank
ma'am.
you.
Ma'am, now you are a co-host, so you
Right.
can share.
Can you see the screen?
Yes,
Yes.
ma'am.
Madam.
Can see.
Right. Okay. So is it time for me to start? About 22 participants are there. Is it all right if I start now?
Okay, ma'am. We'll start now.
Right. Okay, right. So, right. This is a very important topic in microbiology I'm going to do today. And this is on pathogenesis of infections and principles of management of these infections. So I will off my video and continue only with audio, right? So you can concentrate on the text I have shown. Right. So in this lecture, I will do, this is the outline, sources and spread of infection, pathology of infection, clinical effects of microorganisms and host interaction, principles of management, and infection control in practice, right? So these are my topics and the outline today. So we'll take one by one. Let's see sources and spread of infection, how it happens. Now you can see in this diagram, there's a reservoir, immediate source, and the mode of transmission of infection to the host. So the reservoir is the natural habitat for the organism in which it must propagate, right? So, you know, there are several reservoirs. I will elaborate these things in coming lectures. There are reservoirs where the organisms multiply. Okay. So from the reservoir, the organism, sometimes the reservoir could be the immediate source, or sometimes from the reservoir, the organisms can transmit to an immediate host. So an immediate host is a habitat of the organism from which it is transmitted and in which it must survive, right? But may or may not propagate, right? There are some instances the organism propagate or sometimes not. So from the immediate source, the organism come to the susceptible host via several modes of transmission. So now the susceptible host is us, right? Those who get those who get the infection. So there the organism multiply, and we get if the organism wins the battle against the infection. I think there's some disturbance.
Madam, have you logged in from your phone also, ma'am?
Pardon? No, no.
No, two devices? No, ma'am.
No,
Okay.
no. Right. So if the, now it's all right, I think. So if the susceptible host is unable to win the battle done against the organism, then the infection results, right? So let's see how it happens. Now, reservoir. What about the reservoir, right? Sorry, it went away. Where's my right? Do you see it yet? Do you see
Yes,
my
ma'am.
slides?
We can
Do
see
you
the reservoir slide.
Hold on. It has gone. Something has happened. I don't see my slides now, however. Shall I log in again? Please, something has happened.
Ma'am, before logging in,
Uh-huh.
just why don't you try sharing again, ma'am, the document?
Oh, no. Yes, yes.
Yes, ma'am.
I shared again. I shared again, but nothing happened. Shall I log in again?
Okay, ma'am.
Is it
Okay.
all right? Sorry for
Okay,
this.
ma'am. Okay,
Yeah.
ma'am. Okay.
Please make me host again.
Okay, madam. I just made you a co-host.
Thank you. Sorry for the inconvenience. Right.
Can see, ma'am, the reservoir slide now.
Right. The reservoirs are the habitat in which the infectious agent normally lives, grow, and multiply, right? So generally, it could be humans, animals, and the environment. So the reservoir may or may not be the source from which an agent is transferred to a host, as I told you earlier. So sometimes directly from the reservoir the organism transmit, or sometimes it is transferred to an immediate host. So the reservoir of Clostridium botulinum is soil, but the source of most botulism is coming from the canned food. You would have heard about this story. So when the canned food, especially home canned food, is contaminated with Clostridium botulinum spores, so the home canned food tin is the immediate host for the organism, right? So reservoir is not always the immediate reservoir, right? So there are animal reservoirs. So many of the diseases are transmitted from animal to animal. And sometimes humans may be incidental hosts. So we call them as zoonosis. These way of transmitting organisms from animal to animal and human being accidental hosts or incidental hosts, we call them as zoonosis, right? So, and these are some examples for zoonosis: rabies from dogs and other animals, leptospirosis from rats and other mammals we get, brucellosis, cows and pigs, anthrax we get from sheep, plague from rodents, trichinosis from swine, and that is pigs, and tularemia from rabbits, right? So these are the animal reservoirs. So what about environmental reservoirs? Now, as I told you earlier also, plants, soil, and water are reservoirs for some infectious agents. Many fungal agents like histoplasmosis, that is histoplasma is the causative organisms, they generally live and multiply in the soil. And apart from that, causative organism for Legionnaires, that is Legionella multifila, Legionella pneumophila, are found in cooling towers, right? Especially in hospital, even in hospital cooling towers. These air conditioners in cars, they get evaporated and people get respiratory tract infections. And Legionnaires is a very severe disease that happens due to Legionella pneumophila, right? So these are the environmental reservoirs. Now, we human beings also may be reservoirs for some infections. So there we have we are directly transmitting the infection without having any intermediate host, right? So, you know, some diseases like sexually transmitted diseases and viral infections like measles, mumps, and also streptococcal infections, respiratory pathogens like TB. All these are transmitted directly from human to human, and human being the reservoir. So, you know, smallpox is a disease eradicated from the human beings because it was very easy to eradicate because human are the only reservoir for the smallpox. So with that vaccine developed many decades back, it is eradicated now from the world, right? So that is solely because human were the only natural hosts. So the reservoir, right? We know it is a human reservoir. We know it is the source of the infection. So the reservoir may or may not show the effects of illness of the disease. Right. So again, in human reservoirs, patients can be a carrier. Carrier, let's define carrier. Carrier is a person with inapparent infection who is capable of transmitting the pathogen to others. So the person who carries the organism will not show signs and symptoms of the infection, but he is capable of transmitting the infection to others, right? So carriers may be asymptomatic or passive or healthy carriers, right? We call these several names to the same scenario: asymptomatic carrier or passive carrier or healthy carrier. So these are those who never experience the symptoms despite their being infected. So that is very important. Sometimes, say for example, dengue, we are the reservoirs, right? So sometimes we get inapparent disease. We never show symptoms. Incubatory carriers are those who can transmit the agent during the incubation before clinical illness begins. So you would have heard diseases like chickenpox, right? So before appearing of blisters, people can transmit the disease during the incubation period. So at least before 2 days of appearing of symptoms, they can be infectious. And sometimes we have convalescent carriers. Those are who have recovered from the illness but remain capable of transmitting to the others, right? So cytomegalovirus is one of the examples. Even small children who have got cytomegalovirus, they can transmit or shed the infection organism through urine. Then sometimes we have chronic carriers. Those are who continue to harbor pathogens such as viruses like hepatitis B or Salmonella typhi for So now sometimes they can shed the organisms for months or even years after recovering from the infection. So they do not show symptoms but carry. So since they carry for a long time, we call them as chronic carriers. You might wonder why I have given Salmonella typhi like this. It is the scientific way of writing, right? So Salmonella, any organism scientifically writing as the well standard nomenclature, scientific nomenclature. It is with two words. First letter should be in capital and all the others should be in italics. But here Salmonella, the proper name for Salmonella typhi is Salmonella enteritica, subspecies enteritica, and serotype is the typhi. So that is why it is written like this. Okay, right. Then carriers commonly transmit the disease because they do not realize that they are infected, and so they take no special precautions to prevent transmissions, right? So either they don't know or they consider that they have come out from the disease. That is why, right? Symptomatic persons who are aware that they are ill. So they take precautions for others not to get infected. Okay? Or they take treatments to limit the spread. So we have to be careful about the carriers who do not have symptoms, but others now. People do not have symptoms but can transmit the disease are called carriers. People who have symptoms are patients, isn't it? So we have to be very careful considering the carrier state of an infection. Right. So now you have learned about the reservoirs of a disease. Let's see now how it is transmitted. What are the ways of transmitting? So it should transmit from the natural reservoir to a susceptible host, right? There are different ways of transmitting a disease. I told you earlier there are reservoirs. Then some other reservoirs, maybe they are having the organism for a temporary period. Then it is transmitted to a susceptible host. So the transmission is directly to a susceptible host or indirectly to a susceptible host. Direct transmission happens through direct contact or through the droplet spreading. That is direct transmission. Indirect transmission happens airborne way, right? Then it could either be vehicle-borne or vector-borne, right? Airborne, vehicle-borne or vector-borne. So vector-borne also could either be mechanically or biologically. So let's see what these words are and let's clarify how direct and indirect transmission happens. So direct transmission is an infectious agent is transferred from a reservoir to a susceptible host by direct contact or droplet spread, right? So direct contact occurs in these ways. Skin-to-skin contact. So the diseases like impetigo, scabies, herpes happens skin-to-skin, right? Then saliva. Through saliva via kissing, you can get some diseases. Kissing disease or infectious mononucleosis is a very good example for that. And also, diseases like hand, foot, and mouth disease also is transmitted in this way. And we have some other diseases like gonorrhea, which are transmitted through sexual intercourse, right? Then hookworm, leptospirosis-like organisms are transmitted through contact with soil or vegetation, which harbor infectious organisms. That means, if you work in a muddy paddy field, then if it carries leptospirosis, directly it can enter to our system through either mucous membranes or any breach of the skin, right? So hookworms are also, the larvae can enter by penetrating the intact skin. And now, apart from direct contact, we have droplet spread also. It is when you have, get a spray with relatively large, short range respiratory particles, which are produced by sneezing, coughing, or talking of a person who harbors the disease, right? So the droplet spread, you can see, is classified as direct because transmission is by direct spray over very few feet before they drop onto the ground. So, a recent pandemic we had, COVID-19, can be transmitted through this droplet spread. That is a direct method of transmission. And apart from that, pertussis, meningococcal infections, which are transmitted through droplets, influenza, many other diseases can be transmitted through this droplet spread. So now droplet spread is considered as a direct contact because before falling into the ground, if we are very close vicinity with the patient having the disease, directly this spray of droplets can come in contact with our respiratory tract, right? So that is the direct transmission. Let's see the indirect transmission. That is transfer of an infectious agent from a reservoir to a host by suspended air particles, inanimate objects, that is vehicles, or animate intermediates, that is vectors, right? So let's see the airborne transmission first. This occurs when infectious agents are carried by dust or droplet nuclei, right? So then these droplet nuclei can be called as aerosols, which are suspended in air. So generally, these droplet nuclei are, the diameter of this is less than 5 microns. So they can be suspended in air for a longer period. They won't fall to the ground because they are not heavy, right? So they can even take away to a distant area along with the air. They will, the people who are susceptible, even staying somewhat distant away from the patient also can get the disease. So TB, measles, chickenpox are examples for this way of transmission, airborne transmission. So this diagram shows you how airborne transmission happens and the direct transmission through droplets happen. So you can see the red man is an infected person. So the person can produce droplets and airborne particles, aerosols. So you can see ballistic trajectory of the large droplets. That means a normal curve for the large droplets, right? They drop near the infected person, 1 to 2 meter distance. So it is directly coming to the susceptible person, and they can settle on the respiratory tract, right? So the person can get infected. Then the large particles, right? Small particles, they can get included in the air. And if a person stays somewhat away even, can get infected. And apart from this, infected through the respiratory tract, these droplets can deposit on many surfaces. Infected person also can contaminate surfaces, and susceptible hosts can get contaminated their hands while touching these surfaces and get infected because they touch the mouth, nose, and eyes like the mucous membranes and be susceptible for the infection, right? So this is a very good example for that. Right. So from indirect transmission, through vehicles, organisms are transmitted. So the organisms can go to a susceptible host through several vehicles like food, water, and also through biological products like blood, and also through fomites, right? And sometimes through handkerchiefs, beddings, and equipment like surgical scalpels even, right? So through vehicles, the organisms can be transmitted. So this vehicle passively carry a pathogen, as food or water carry hepatitis A viruses, right? So apart from that, the vehicle may provide an environment in which the agent grows, multiplies, or produces toxins sometimes. So improperly canned foods provide an environment to propagate and produce toxins from Clostridium botulinum, right? Likewise, the environment can provide opportunity to multiply, or this vehicle may passively carry the pathogen to a susceptible host, right? So this is indirect transmission through vehicles. What about vectors? Like mosquitoes, fleas, ticks can carry infectious agents through by mechanical means or by supporting the growth of organisms, right? So what are the examples for mechanical transmission? Fleas carry Shigella on their appendages. So, when they sit on excreta and then can contaminate uncovered food, right? So through their legs, they transmit Shigella. Flea can carry Yersinia pestis, the causative organism of plague, right? In their gut. So mechanically, without causing any change to the organism, they can transmit the organism. What about biologic transmission? That is, the causative agent of malaria undergo maturation in the intermediate host before it can be transmitted to humans. So here, the malaria parasite undergoes changes. Without undergoing changes, it cannot cause disease in humans. Same applies to dengue virus. After a feed from an infected person, the dengue virus should undergo a cycle in Aedes mosquito's gut, right? So without going that transition period, it cannot affect another susceptible host, right? So that is biologic transmission. So this table shows you some reservoirs, sources, and modes of transmission. So you can see the infective disease, the agent, the reservoir, intermediate source, and mode of transmission. If you take sore throat, the causative organism is Streptococcus pyogenes. Reservoir is the human upper respiratory tract. So the immediate source is a human upper respiratory tract, and patients get this disease through from another person. So we call them as exogenous infection. When they get the disease from another person, we call it as an exogenous infection, and it is acquired through the airborne droplets. What about oral thrush? Oral thrush happens due to Candida albicans, and the reservoir is most human mucosal surfaces. In many mucosal surfaces, you get this Candida albicans. In mouth, right? Sorry, in gastrointestinal tract, in genital tracts, you get, and even on skin, you get Candida albicans. So the immediate source is normal microbiota of oral mucosa. So it is totally endogenous. So when you get antibiotics, right, your normal flora is suppressed and Candida albicans can get overgrown. So you can get infections due to Candida albicans, that is oral thrush. And apart from that, immunosuppressed patients, their immunity is low, they cannot combat or they cannot fight with normal commensal flora, and the albicans, even being a normal commensal flora, they can cause oral thrush, right? Let's see about tetanus. Tetanus, you know, the bacterium Clostridium tetani is the positive agent, and reservoir is soil or animal intestine, including us. So any environmental contamination with soil or animal feces, a person can get the infection, tetanus. It is totally exogenous. So if you get especially a penetrating injury, tetanus is a likely infection. Why do we think about penetrating injuries? You know, Clostridium tetani is an anaerobic organism. So deep wounds, in the deep wounds, you do not get oxygen adequately to kill the organism. So this anaerobic environment favors the growth of Clostridium tetani. Syphilis. Syphilis is caused by Treponema pallidum. Human beings are the reservoir, infected human beings. So the immediate source is patient with genital ulcers or secondary syphilis. These people are highly infectious. So through sexual contacts, people can get syphilis. So it is an exogenous infection. What about yellow fever? Yellow fever virus is the causative agent, and the normal reservoirs are monkeys. So infected human being or monkeys are immediate sources, and through mosquitoes, exogenously, this infection is acquired. Now you know yellow fever is transmitted through Aedes mosquitoes, right? So how many diseases, severe diseases are caused by Aedes? You know, dengue, chikungunya, yellow fever, and even Zika virus, right? So all these are caused by a single type of Aedes mosquitoes, not single type of species. There are Aedes aegypti, Aedes albopictus. These are the vehicles, right? Now, in fact, not vehicles, vectors, right? So, AIDS, acquired immunodeficiency syndrome, you know, the causative agent is human immunodeficiency virus. So, infected human beings are the reservoirs, but through human blood, sexual contact, right, can be the way of transmission. So, bloodborne or sexual contact are the modes of transmission. Toxoplasmosis is a parasitic infection. Organism is Toxoplasma gondii, and the reservoir is cats. Reservoirs are cats. So, if somebody takes undercooked meat or contact with areas contaminated by cat feces, you can get the infection. So, exogenous infection through ingestion, we get the disease, right? So, this table gives a summary of overall modes of transmission and different infections acquiring through reservoirs or immediate sources. Right. So now you know many things about the diseases, and let's see what are the portals of entry of the infections. So the portal of entry refers to the manner in which the pathogen entered to a susceptible host, right? So the portal of entry must provide access to tissues in which the pathogen can multiply or a toxin can act, right? So generally, infectious agent use the same portal to enter a new host that they use to exit the source host. You know, generally, say I am infected from a disease, say influenza. So I excrete organisms through respiratory tract via coughing and sneezing. So this existing source host is through respiratory tract, and it is again acquired to another patient through the respiratory route, right? So in contrast, some pathogens that cause gastroenteritis follow fecal-oral route because they exit the source host in feces but are carried inside the body through food, water, or utensils that are contaminated with the causative organism. So they enter through the mouth, but same gastrointestinal tract. Okay. So other portals of entry are skin, you know, hookworm, leptospirosis, enter through skin, and mucous membranes, so syphilis, gonorrhea, enter through mucous membranes, and through blood like hepatitis B, human immunodeficiency virus, hepatitis C, all these enter through blood. So this shows some ways of entry, airborne, right? You can see Mycobacterium TB, influenza, parainfluenza virus, rhinovirus, respiratory syncytial virus, varicella zoster virus. Aspergillus fumigatus, airborne spores, Legionella pneumophila, all these enter through airborne routes. What are the fecal-oral methods? Viruses causing gastroenteritis like norovirus and Clostridium difficile even, right? Spores enter through mouth. Salmonella enteritis, vancomycin resistant enterococci, These enter through femoral road. Skin-to-skin contact, MRSA, Streptococcus pyogenes. VRE also can enter skin-to-skin. BL producing polyforms, right? Then CPE, multi-resistant Acinetobacter, right? So indirect contact, norovirus, influenza, MRSA, Streptococcus pyogenes. Likewise, you have a lot of long list. Bloodborne, HIV, HBV, HCV, through sharp injuries, blood of fluid splashes onto the mucous membranes, then waterborne diseases, Pseudomonas aeruginosa, you can see them even in tap water, Legionella pneumophila. Then reusable surgical equipment sterilized by standard procedures. Now it's very rare, Creutzfeldt-Jakob disease in our country, right? But in developed countries, they have this disease because, prion diseases, this organism called prion, or organelle called prion, is very difficult to get rid of through sterilization. You have to have requirements 3 times more than the normal sterilization. Okay. So these are the ways of acquiring the diseases. So let's see how a pathogen enter and exit from the body. To establish an infection in the host, pathogens must attached across the body surfaces. Many pathogens have developed chemical or mechanical mechanisms to attach themselves to the surface of the respiratory, urogenital, or any tract in the body, right? So, for example, in the skin, they generally depend upon the entry via small wounds or arthropod bites. So, pathogens must exit from the body after replication in order to be transmitted through fresh hosts, right? So, this takes place across body surfaces. Sometimes you can see shedding of pathogens from the respiratory or skin or urogenital or alimentary tract, and delivery into the blood or dermal tissues for uptake during arthropod feeding. So that is how they exit. I think you have got the idea, isn't it? So they can enter the organisms as well as exit from the host. So many human infections come from animals, either directly as zoonosis or indirectly via blood-sucking arthropods. And the incidence of these infections depend upon the exposure to infected animals or arthropods, right? That is why we have to prevent getting exposed to these vectors. Right. So how an infection settle or initiate in a susceptible host? People get infectious diseases when microorganisms overpower the host defenses. That means you get the balance between organism and the host shift in favor of the organism. There are 2 critical determinants in overpowering the host. So, one is the number of organisms to which the host is exposed and the virulence of these organisms. Greater the number of organisms, the greater the likelihood of infection, right? So, over a small number of highly virulent organisms can cause disease, just as a large number of less virulent organisms can. Right? So I told you, number and virulence are the most important things. Higher the number of organisms enter, higher the possibility of getting infected. But sometimes even with low number, the highly virulent organisms can cause the infection. So let's see about the defense mechanisms, right? So non-specific defenses of the body rely on anatomical, physiological, and bacteriological microbiome components. Now, these defense mechanisms are very important to prevent settling of the infection. So, you know very well about these things. Ciliated epithelium of the respiratory tract. It pushes mucus containing trapped bacteria out of the lungs. Right? Either these are swallowed or destroyed by the gastric juices. Then lysozyme. It is an enzyme in the saliva. It degrades the peptidoglycan components of the bacterial cell walls. Then macrophages of the spleen, lungs, liver, they clear small number of bacteria coming into the bloodstream. Then the low pH of the stomach, they kill many microorganisms that are swallowed. Bladder, the frequent flushing of urine prevents bacteria settling or ascending from the urethra to the bladder and kidneys. And the normal flora of the bowel, they have many anaerobes. They create colonization resistance of other pathogenic bacteria. Then skin. Intact skin is an excellent barrier for infection. You know, first few layers are non-live layers, right? They create a good barrier, prevent organisms entering into the body. So this is very important in combating infections, and they are considered as first-line mechanisms. So, as I told you, normal flora, they again are called as commensals, right? So, various bacteria and fungi are the permanent residents of the body. Some body sites, especially skin, oropharynx, colon, vagina, right? So, these are the normal flora. And they create some resistant mechanisms, inhibitory substances, and also they compete with other pathogenic organisms. Viruses and parasites, such as protozoa and helminths, are usually not considered as members of the normal flora, although they can present in asymptomatic individuals. These organisms drive benefits, the normal flora, from the host, but do not damage the host. The term human microbiome is often used to describe the normal flora, right? Human microbiome. You have heard about the term carrier. It is that an individual harbors potential pathogens and therefore can be a source of infection to others. I'm going to let you know another word, that is colonization. It is typically refers to acquisition of a new organism, right? Acquisition of a new organism. So after the new organism colonizes, that means attaches and grows, especially in mucous membrane, it may cause an infectious disease or it may be eliminated by the host defenses. Furthermore, the person colonized by a new organism can transmit the organism to others. That means the person can act as a reservoir of the infection for others. So colonization is an important phenomenon that means harboring a new organism. It allows to multiply and settle down, and also can be as a source or reservoir causing infection to others. Okay, right. So the members of the normal flora play a role both in the maintenance of health and in the causation of the disease. I told you they constitute a protective first defense mechanism. The non-pathogenic resident bacteria occupy attachment sites on the skin and mucosa and can interfere with the colonization by pathogenic bacteria. Right? So normal flora interfere with colonization by pathogenic bacteria. So if the normal flora is suppressed, pathogens may grow and cause disease. What are the instances we suppress normal flora? By taking antibiotics. So antibiotics can reduce the normal flora and allow the growth of some organisms such as Clostridium difficile, right? Candida, like organisms. If the Clostridium difficile is overgrown, it is resistant to antibiotics because they are spore forming. So they can cause a severe condition called pseudomembranous colitis, which is an antibiotic-associated diarrhea. So here, the organism not only cause diarrhea, but there will be a pseudomembrane and causing colitis as well. Right? So this can be, if the patient is not treated, this can even be fatal. So apart from that, the normal flora can cause disease, especially in immunocompromised and debilitated individuals. Although these organisms are non-pathogens, in their usual anatomic location, they can be pathogens in other parts of the body. And also, the normal flora may serve as a nutritional function. These intestinal bacteria can produce vitamin B's and vitamin K's. So poorly nourished people who are treated with oral antibiotics can have vitamin deficiencies because along with oral antibiotics, the normal flora is destroyed. So they should be supplemented with vitamins, right? However, we have to consider that normal flora is not essential for proper nutrition of the patient. Okay, right. Right. So we now considered about defense, primary defense mechanism. Now, apart from that, we have active defenses also in the body. This active defense system is based on the immune responses and its ability to differentiate self from non-self and identify and destroy invading microorganisms. So we have two main mechanisms, antibody response and cell-mediated immunity. Right. So then let's see what are the principles of pathogenesis of these organisms or infections, right? So a microorganism is a pathogen if it is capable of causing disease. Some microorganisms are highly pathogenic. You know that. That means they can cause disease, whereas others cause diseases rare. Opportunistic pathogens are those that are rarely cause disease in immunocompetent people, but can cause serious infection in immunocompromised patients. These opportunistic pathogens are frequent members of body's normal flora. And they have the ability to take the opportunity offered by reduced host defenses to cause diseases. Right? So, if the host has reduced defenses due to some disease, then the opportunistic pathogens come into play. Right. So, virulence is main factor in pathogenicity. Virulence is a quantitative measure of pathogenicity and is measured by the number of organisms required to cause disease. Right? So this bit may be somewhat boring, but please bear with me. So virulence is measured. The 50% lethal dose is again called as LD50, right? Is the number of organisms needed to kill half of the hosts. Say some organism group of organisms, right? Say the same organism, the number of organisms need to kill a heap of peoples, right? So 50% of lethal dose is the number of organisms needed to kill the half of the host. And 50% of infectious dose, ID50, is the number of organisms needed to cause infection in the half of the host. Right? So organisms with lower LD50 or lower ID50 are more virulent. That is, low number of needed to kill or infect people. Right? So lower LD50 or lower ID50 are more virulent than those with higher LD50 or higher ID50 because fewer organisms are needed. Right? So the virulence of the organism is determined by its ability to produce various virulence factors. So the organisms have various virulent factors. They have villi to get adhered to a mucous membrane. They can produce exotoxins or endotoxins. Some of them possess capsules to protect them getting phagocytosis. And some can survive various non-specific host defenses, such as acid environment in the stomach, right? So these are some of the virulent factors of the organisms. So the infectious dose, the term infectious dose is of an organism required to cause disease varies greatly among the pathogenic bacteria. If you take Shigella and Salmonella, they both can cause diarrhea by infecting gastrointestinal tract. But the dose of Shigella, infectious dose of Shigella is less than 100 organisms. Whereas infectious dose of Salmonella is more than 1 lakh, right? So you can see Shigella has low LD50 and low ID50 than Salmonella, and Shigella is more virulent. Okay. So bacteria cause disease by 2 major mechanisms, right? While initiating an infection, they can invade and form inflammation, or they can produce toxins, right? So invasive bacteria grow to large numbers locally and induce an inflammatory response consisting of erythema, edema, warmth, and pain, giving rise to symptoms and signs of the disease. That is invasive bacteria. Now, toxin-producing bacteria. Now, these bacteria can produce two types of toxins. One is exotoxins and the other one is enterotoxins. Exotoxins are polypeptides. Remember, polypeptides released by bacterial cells. But endotoxins are lipopolysaccharides. Lipopolysaccharides, which form an integral part of the bacterial cell wall. So, you know, bacterial cell wall, only gram-negative bacterial cell wall have lipopolysaccharides. So, exotoxins occur only in gram-negative bacteria. And it causes fever, shock, and other generalized symptoms. So, these both exotoxins and endotoxins by themselves can cause symptoms when the presence of bacteria in the host is not required. Right? So, now say just one bacteria or so can produce toxins, and that toxins per se can cause the disease. The presence of bacteria thereafter is not needed to cause symptoms and signs of the disease. Right? So you can see that invasion and inflammation is one way and toxin production is one way. So out of toxin production also, you have endotoxin production and exotoxin production. So endotoxin is only produced by gram-negative bacteria because it is a part of the bacterial cell wall, lipopolysaccharide. But exotoxin is produced by both types of bacteria. Right. So endotoxins, bacteria like E. coli, Neisseria meningitis can initiate endotoxin shock. With the outer membrane of gram-negative bacteria, you have lipopolysaccharide endotoxin, which can activate the alternative complement pathway. So when the alternative complement pathway is activated, the result is membrane attack complex that release endotoxin, which leads to activation of large number of macrophages, causing uncontrolled release of cytokines, giving rise to endotoxic or septic shock. Right. So what are these super antigens? You would have heard about these super antigens. Now, some bacteria can produce potent exotoxins that act as super antigens, which cause macrophages and T cells to interact in a non-specific manner. So you can see all these immune cells are activated in a non-specific manner. This results in massive cytokine release leading to shock. So generally, people go into septic shock partly due to our overall immune activation, right? So our body's immune system when overtly activated, people can go into shock. So some examples are septic shock, toxic shock syndrome, which is caused by Streptococcus pyogenes or Staphylococcus aureus. There they produce a toxin called pyrogenic toxin. Both the organisms can produce this toxin. Here, the macrophages and T cells get activated, right? So this pyrogenic toxin is a super antigen. Right. So apart from these, right, pathogenic mechanisms, some people get immune-mediated diseases as well. There, if a person gets recurrent pharyngitis untreated due to Streptococcus pyogenes, the person may result in activation of an immune response to the organism, which cause damage to the host's own tissues, including heart valves. So the basis of rheumatic fever is this. In rheumatic fever, because of the organisms, because of the antibodies produced by the organism, right, produced due to the organism, it gets molecular mimicry to body's own tissues. So it attacks the heart valves, joints, skin, and even central nervous system. That's why people get rheumatic heart disease, rheumatic arthritis, rheumatic nodules in the skin, and rheumatic chorea, right? So it is an immune-mediated disease, and it is a sequelae, non-suppurative sequelae of Streptococcus pyogenes, right? So these are some pathogenic mechanisms. Now, apart from that, there's another scenario, right, called biofilm formation. So bacteria and some fungi can cause biofilm. So biofilm is an aggregate of interactive bacteria adhere to a solid surface. Adhere to the solid surface, attaching to each other and encased in an exopolysaccharide matrix. This is distinct from planktonic or free-living bacteria, in which interactions of the microorganisms do not occur in the same way. So biofilm forms a slimy coat on solid surfaces and occur throughout nature. Now, say if we insert a catheter to a patient, inside the catheter lumen or even outside the catheter lumen, bacteria can form a slimy coat called biofilm. Even IV cannulas are affected from biofilms. That is a small coat of organisms, where the specific nature is the organisms are embedded in a solid, embedded in a exopolysaccharide matrix. I'll show you a picture. So a single species of bacteria may be involved, or more than one species may aggregate to form a biofilm, generally a single species. Fungi are occasionally involved. Bacteria in the exopolysaccharide matrix may be protected from the host immune mechanisms, right? So host immune mechanisms cannot act because bacteria are inside the matrix. This matrix also functions as a diffusion barrier to some antimicrobials. So bacteria within the biofilm show marked resistance to antimicrobials compared with the same strains of free-living bacteria, right? So this explains why it is so difficult to treat infections associated with biofilms. Biofilms are important in human infections that are persistent and difficult to treat. I will give you an example, Staphylococcus epidermidis. So you know, in central venous catheters, Staphylococcus epidermidis is a skin commensal, right? They go and form a biofilm in central venous catheters, and it is a well-known fact. And also in prosthetic joints and also in cardiac device infections. Pseudomonas aeruginosa are airway infections in cystic fibrosis patients, and also in eye infections with contact lenses. dental plaques. These are very common instances where you get biofilm formation with these organisms. So this is a picture of biofilm formation. So you can see the bacteria, planktonic, that means free-living bacteria, come and attach to a solid surface. And this attachment is reversible. But when, after some time, they get irreversible attachment when they produce that extra polysaccharide matrix substances. So you will see a cluster of cells protected inside this matrix. And then they produce a bigger mass called microcolonies. Right? They grow bigger and bigger, and they mature bigger and bigger, and then after some times they can disperse free-living bacteria into the circulation. So this bacteria can go and settle and produce a biofilm in another site of the body as well. Right, so let's see about the viruses. Viruses are completely dependent on the host for their replication. So they are obligate intracellular pathogens. They cannot survive outside the animal or human hosts. Right? Whatever hosts outside the host cells. So they have a lot of appendages in viruses. So a part of the capsid, that means in case of non-enveloped viruses, or the enveloped binds to a specific receptor or receptors in the host cell to initiate the entry of the virus into the cell. This interaction of virus with cells can result production of new virus particles with or without lysis of the host cells, abortive infection, latency, or transformation. I will let you know what these words are. First of all, I thought of letting you know about the parts of a normal virus. You can see the outermost part is the envelope. And sometimes you can have projections, that means envelope proteins of virus. Then inside the envelope, you have the capsid and the viral genome and enzyme. So it's very basic. This is the basic structure of the virus. Right. So viruses that infect and replicate within the cells, causing the cells to lysis when the progeny virus is released, right? So this is called cytolytic cycle. So virus infect the cells and the host cells will get lysed. So it is cytolytic cycle. The infection is productive and the cell culture demonstrate cytopathic effects, which are often characteristic of the infecting virus. So cytopathic effects you can see from microscopes, right? Although you can't see the virus under light microscope, cytopathic effects can be seen because we look at the cells of the host. So these host cells are termed permissive cells. That means the cell permit to infection continue and the cells are lysed due to the infection. But in some instances, viruses are produced from the infected cells, but the cells are not killed by the process. So the infection is productive, but non-cytolytic cycle is going on and may become persistent. That means continuing, right? So some viruses that infect cells but do not complete the replication cycle. They are non-productive, right? So the infection is called abortive. Abortive infections can be due to mutations in the virus, so that some essential function is lost, or to the production of defective interfering particles, or to the action of interferons. That is a protective mechanism of the host. So now you know about cytolytic cycle with permissive host cells, non-cytolytic cycle with persistent infection, and abortive or non-productive cycles. Sorry. Some viruses enter the cells but are not produced by the infected cells. They enter the cell but not produced by the infected cell. The virus is maintained within the cell in the form of DNA, which replicates in association with the host cell DNA. So the host cells termed non-permissive and the infection is non-productive, right? Non-permissive, non-productive. But occasionally, this type of interaction results in transformation. What is transformation? Turning the cell into a tumor cell. It is called transformation. Right. In some cases, latency happens where the virus persists as its genome with the limited expression of the selected viral genes, sometimes as RNA only. So in latency, no symptoms happens, but limited expressions happen. And after some times, the virus, if the environment is favorable for the virus, it can cause the infection. But this silent period is called latency. So you can see the viral replication cycle. One is the virus. Second one is attaching to the surface of a susceptible host. You can see. So third one is entering into the host cell with various mechanisms. So fourth one is uncoating, right? This capsule and the envelope, and the for viruses we call it as an envelope. Envelope and the capsomeres are released or removed. Then you can see 5 undergoing early transcription and translation. 6 replication of the viral nucleic cells along with the help of host cells, host nucleic DNA and RNA, right? Then 7 is 6 is replication of the viral nucleic acid. 7 is late transcription and translation. 8 is assembly. So it gets assembled with all the viral coatings and then released from the cell. Right. Let's see how the body reacts to an infection. The acute reaction. So antibodies start to appear after 4 to 5 days of the infection. So immune system recognizes the organisms as foreign. So that is the innate immune system starts activation immediately, right? Then the other arms of the immune defense, innate immunity, are of critical importance fighting an infection in the early stages. So, macrophages, even T cells initiate the cytokine release. The cytokines involved include tumor necrosis factor, gamma interferon, interleukin 1. So, being distributed throughout the body, cytokines coordinate the acute response to infection and stimulate many organs of the body. So, you can see the interferons, interleukins, gamma interferon, tumor necrosis factor, interleukin 1, interleukin 6. So, in acute response, hypothalamus start fever, brain starts somnolence, that is a state of drowsiness, sleepiness, or strong desire to sleep. Vascular permeability changes, giving rise to hypotension and shock. Sometimes mobilization of WBC from bone marrow cause neutrophilia, right? And cytokines such as interleukin 6 stimulate the liver and release of acute phase proteins from the liver. So you will get high CRP readings from patients, and they have high ESR readings also. Mannose binding lectin binds to the mannose residues, and as phagocytic cells have receptors for this bound mannose binding lectin, the bacteria are more readily taken up. So it is termed opsonization. So in the acute stage of infection, before any antibodies are present, the alternative complement pathways can also be activated. When IgG antibodies bind to antigen, such as that on the surface of viral infected cells, the classic component cascade is also activated. So you can see before the specific immunity activates, many components of the innate immunity activate and start to fight with the organism. So the effect of prolonged cytokine stimulation, which occurs in chronic infections such as TB or bacterial endocarditis, are characterized by weight loss, right? And low serum albumin, representing the outcome of cytokine-stimulated catabolic process. That is why you see a weight loss in such chronically infected patients. Right. So, let's see how infections is managed, the principles. Here, the key strategies to consider when managing a patient with infection and the severity of the situation will determine the sequence of events. Right? So, key strategies are there. Severity will determine the sequence of infection. So, when you are assessing a patient first, you have to assess about the history and you have to do the examination. Right? So, along with that, investigations, microbiology, hematology, histopathology, immunology, and imaging studies are done. And all three together, you can do the diagnosis or differential diagnosis at any stage from the initial assessment. And it depends on the clinical, laboratory, and imaging findings. So there you can see what is the likely diagnosis of differential diagnosis, which organism is likely to be involved, and have the correct specimen sent, right? Have previous treatment and infection control alerts been checked? So like resistant organisms you can consider, right? And has the correct antibiotic treatment been prescribed and given? So along with that, you can have an action plan which along with the differential diagnosis is documented, right? So confirm the necessary specimen have been collected and that the antibiotics has been prescribed and the first dose given. What further tests or referrals are needed to confirm the diagnosis of this patient, especially as GPs, it's very important for you. Is there a clear plan to review the next subsequent doses of antibiotics based on the renal and liver function tests? Have the necessary infection control alerts been actioned? And have the necessary public health alerts been actioned? So you have to consider everything when you are planning, managing these infectious diseases. Okay, so let's come to a very important part, microbiological diagnosis, if you have a patient with infectious diseases. I'm asking you, do you want a break now? Anybody?
No, madam.
No. Okay, right. Let's continue there. So then, in microbiological diagnosis, it's very important to identify the etiological agent. So all the microbiological diagnoses are done in view of identifying the etiological agent. So for that, we have to have good quality specimens, right? Good quality specimens are needed for reliable microbiological diagnosis. Microbiological confirmation of a clinical diagnosis of infection depends upon the collection of high quality specimens and their rapid dispatch to the laboratory with all the necessary supporting information, right? So it is a must. Laboratory tests detect microorganisms or their products or evidence of patient's immune response to infection. Apart from normal conventional investigations, we have newer molecular techniques also now. That is PCR, mass spectrometry, right? They are now, even in Sri Lanka, are increasingly used to detect pathogens rapidly. However, ABST is very important in treatment, which can only be done with culture isolation. Okay. So, for conventional microbiological investigations, to grow bacteria take at least 18 hours. Generally, we do not do isolation of viruses, right? But we do isolate fungi. Standard culture results cannot be expected in less than 24 hours, right? So interpretation of culture results depend upon the source of the specimen, however. So generally, from sites which are normally sterile, we consider any organism isolated from that site as significant. Sterile sites, most of our body sites are inside body sites are sterile, right? So CSF, blood, even urine you have to consider as sterile, but urine is collected through perineum. Okay, so there we have a formula to consider which specimen is which organism is pathogenic, right? So it's very difficult for us to determine which is the pathogenic organism from sites, especially from sputum, from feces, right? However, we, with our knowledge and with the patient's history, we have to determine which pathogen is, which organism is the pathogen. In fact, from feces, it is straightforward sort of, because Salmonella and Shigella are not commensals, but from sputum, it is and from wounds and wound swabs is somewhat difficult. So good. However, that's why good communication between the clinician and the microbiologist is extremely important. And always we expect brief history of a patient, and also the antibiotics the patient had been on. These things are very important for us to interpret these pathogens, right? So specimen collection and transport is very important. Now, collect the appropriate specimen from the correct site when you are getting specimen from a patient. That means blood and cerebrospinal fluid from a patient suspected of having meningitis should be collected. Then collect the specimen at the appropriate time. So you have to collect the specimen during the acute phase of illness. For culture, for antigen detection, and for PCR, you have to collect in the acute phase. But antibody detection can be done later because it takes time for the patient to develop antibodies, at least about 7 days. So, however, in the initial stage, you have to collect the specimens as soon as possible when the patient is sick. And also, if possible, collect specimen before patient receives antibiotics. That is very, very important, right? Otherwise, the existing organism will not be adequate for us to culture. Now, generally, especially if I tell you about TB, for us to see in acid-fast bacillus smear, there should be about 10,000 organisms per ml to visualize in the smear, right? So that is why before starting antibiotics, it's good to send a specimen. Collect enough material and adequate number of samples. For example, if a patient is having infective endocarditis, you have to collect at least 3 sets of blood cultures, right? When you are sending a wound swab, send 2 swabs. 1 for culture and 1 for gram stain. It is important. Avoid contamination from normal flora or external sources, right? So as soon as the specimen is collected, say you are collecting a throat swab, then swab the throat area only. Do not touch the swab with tongue, the teeth or anything, right? It should be the tonsil area only. And use the correct containers and appropriate transport media. So containers should be sterile, apart for feces and for sputum for AFB, right? All the other containers for microbiological investigations should be sterile. And give proper instructions to the patient. So if you are asking the patient to collect a urine sample, you have to give proper instruction. Sputum samples, yes, because the patient collects those samples, right? So when collecting a midstream sample, then you have to ask the patient to wash the area first and then wipe if possible, then pass the first part of urine to the toilet, then collect the middle part, and then close the lid of the container promptly without washing it. Likewise, you have to advise. Then, when you are sending the specimens, label it properly, complete the request form with necessary clinical information and statement of possible etiology, and your suspicion, say whatever the DD you have, right? It will be easy for us. Then inform the laboratory if special tests are required. Okay, so for example, if you send a sample of feces for culture, then, and if you are looking for Campylobacter also, you have to request. Otherwise, Campylobacter needs special culture media. So we do not do that if you do not request. Okay? And transport specimens rapidly to the laboratory under appropriate conditions. So you can see, if you send a good specimen, you will get a good report. But if you get a garbage, right, then the result will be also garbage, right? So remember to send a very good specimen for microbiological investigations. So microbiological diagnosis of infection can be performed in several ways, right? So when we receive a specimen, we can do several steps in testing the specimen. One is direct detection of microorganisms by microscopy with or without staining. Second one is identification of microorganisms by isolation and culture. Right? We can do that. I will elaborate one by one later. Then identification of a specific microbial component. We can detect antigen of the organism or toxin. Then detection of specific antibodies to a pathogen in patient serum. We call it as serology. Then after that, detection of specific nucleic acid of the organism. So for that, we do molecular diagnosis. So what about direct examination? There we do microscopic evaluation with or without stain, right? So the consistency of the specimen is very important. So here, rice water stools for cholera. There you can see, if you put a drop of watery stools to a slide and look under the microscope, you will see moving bacteria. Then visible parasites in stools, hemolysis of blood, right? Turbid urine, we can see under the microscope. Sorry, under the macroscope, that is macroscopic evaluation, right? So that is without a microscope, you just look at the don't mention about what I told earlier. This is a macroscopic evaluation. You look for the consistencies. For example, stools. You can see watery stools or mucoid stools, right? Or stools with blood and mucus. And for cholera, you will see the rice water appearance. Now, you would have heard about rice water appearance of cholera stools, right? So, and sometimes you can see visible parasite worms in stools, right? Then blood got hemolyzed and urine is turbid. So that is the macroscopic appearance, and it is very important, and you have to report it even in the report you issue. Then you can do direct microscopy, right? So wet mount techniques we do. Now here, you can see in the picture, you can see a wet mount of nail scraping, right? So there you can see budding yeast. Likewise, you can do a wet mount. And you can do a hanging drop method. They are just rice water stools. There you can see moving bacteria, cholera bacteria. Then you can see white blood cells, that means pus cells in the specimen. That means there's inflammation. Sometimes you can do dark brown microscopy for fragile organisms like spirochetes. That means for leptospira, for syphilis, you can see this dark brown microscope. Now, parasites variably maintain. Some in parasites, protozoa, helminths, eggs, you can see, right? and also moving bacteria in the specimens. So that is direct microscopy. And electron microscopy is also important, especially for viruses. And then we can do staining and look under the microscope. So Gram staining, Zeal-Nielsen staining for mycobacteria, modified Zeal-Nielsen staining for leprosy, cryptosporidium we can do. Then specific staining with labeled antibodies. So we do immunofluorescence. It is used when Gram stain cannot help in diagnosis, like in cases of rickettsia, right? So where we cannot do Gram staining, that means the organism is too small to be visualized in Gram staining, we do. Okay, so that is special staining with labeled antibodies. And also we can do rapid tests. That is, bacterial, viral, or parasitic antigens can be detected by this rapid test. Toxins in biological fluids like CSF, blood, urine, we can do. And the techniques are direct agglutination, that means slides or cards, latex agglutination, and immunochromatography, that means dipstick. So with all these, we can detect the organisms. That is direct examination. So I want to elaborate more about microscopy. So it is the important first step in the examination of most specimens. And in certain situations, it will guide the management of patients as well. So for example, if we see gram-stained organisms in CSF or incubated blood culture, right, or any sterile fluid, in patients, it will guide the selection of antibiotics until the ABST is available. Now, for example, in CSF specimens, we do gram stain and see gram-positive cocci in diplococci. So now we know what are the common organisms causing meningitis in adults, right? So then we know that this could be Streptococcus pneumoniae. So before the ABST is available, it takes another 2 days. We can inform the clinicians that these we can solve, and you better start treatment according to this. So the patient will survive because he has got proper antibiotics 2 days before the ABST reports available. Okay. Gram stain of a smear made of urethral discharge showing gram-negative intracellular diplococci enables commencing treatment for gonorrhea. So you know in urethral discharge, if you see gram-negative diplococci, that means intracellular diplococci, that is very characteristic of Neisseria gonorrhoeae. So you can start the treatment nearly by gram stain. And again, acid-fast bacilli with typical morphology in a smear made of sputum from a patient with pulmonary tuberculosis. So then you know this patient is positive for FB, and then you can start treatment. And in some instances, especially in fungal infections, in KOH, you know, when we get a scraping of, say, nails, superficial skin scraping, then you can put a drop of potassium hydroxide and look under the microscope. There you see filaments. Then you can start treatment. Okay. And in some instances, in herpes simplex virus 2 infection, patients present with ulcers, blisters, and they go into ulcers next. So if you get a scraping from that ulcer and do this Giemsa stain, you will see giant cells, right? Similarly, Negri bodies, that is, if a rabid dog's brain is taken, you can do a special staining called cellar staining. There you get specific inclusion bodies of infected cells of the dog's brain. Those are called Negri bodies. This will give a specific clue for rabies in the dog. So you can start vaccination of this patient. Right? So likewise, it's very useful. The microscopy is very useful. So we combine all these techniques together to diagnose an infection. So similarly, electron microscopy is used to visualize the morphology of viruses. So I have given you some examples of microscopy. So first picture is gram-positive diplococci, similar to pneumococci. Again, you can see gram-negative bacilli. A top third picture is yeast, how candida is appearing in gram stain. Then the second row left, you can see intracellular gram-negative diplococci, suggestive of gonococci, right? So second row, right hand, Moraxella catarrhalis is there. You can see gram-negative diplococci outside, basically outside the pus cells. Then the last picture is nice curved, fine, red-colored bacilli in blue background. It is the acid-fast stain or Ziehl-Neelsen stain, where you get Mycobacterium TB in appearing, giving the characteristic features, right? Right. So we can do fluorescent microscopy also. The advantages here are high sensitivity and specificity, rapid testing, and easiness to perform. So you can see the apple green parts are the positive parts. There are respiratory cells, and fluorescent antibodies are tagged with that. So the infected cells are appearing as apple green color. This is a respiratory specimen where you can detect the different organisms, viruses from this method. Right. So we have come to isolation of the organism by culture. You can see here we do isolation in artificial media. So you can do bacteria and fungi isolation in artificial media. But viruses, they do not grow on artificial media. So we have to have cell lines, right? But some bacteria, like Treponema pallidum, which causes syphilis, and Mycobacterium leprae, which causes leprosy, cannot be cultured in artificial media, right? And even not in cell lines. So they are maintained in lab animals. So some viruses also can be maintained in lab animals. So why do we do culture? To identify the causative agent, we have to do culture. And also to differentiate pathogens from commensals, we do culture. So after doing culture, we can we can look for the proper characteristics of the pathogen, and then we can come to a conclusion. And also, if we say now sometimes in blood cultures, we can get coagulase negative Staphylococcus. You know, coagulase negative Staphylococcus is a commensal. But if you get in repeated cultures the same organism and still the patient is sick, then we can come to a conclusion that coagulase negative pathogen Staphylococcus is the pathogen here. So that is also important. And to determine antibiotic sensitivity of the pathogen, we do this culture. Apart from that, to obtain a colony count for determining the clinical significance, we do culture. Now, especially urine, we do interpret urine culture according to the colony count, number of colony count. So if we get more than 10 to the power 5 colonies per ml, it is the significant colony count, right? So for that also we do culture. And for epidemiological purposes, cultures can be done. Now, for example, if there's an outbreak going on, say a diarrheal outbreak, so if we isolate the same organism from all patients affected, and when we type them and coming into the same strain, then we can say that it is an outbreak happening from the same source, right? So for such epidemiological purposes, we do culture. So you can see some examples of culture. We use artificial culture media. The first picture is a blood culture plate. You can see Streptococcus pyogenes growing in blood culture, tiny colonies, hemolytic colonies. The second picture is McConkey agar, which is again used routinely in our laboratory. The third one is chocolate agar. So these blood agar, McConkey agar, and chocolate agar are used generally in routine practice in our laboratory. The second row, left hand, is CLED agar, cysteine lactose electrolyte deficient agar, which is used to process urine samples, right? It is sort of a green colored medium, which gives yellow colored colonies and colorless colonies in different organisms. The middle one is Salmonella shigella agar. There you can see, sorry, it's an XLD agar, where you have salmonellas, where we used to plate salmonellas, that means feces. We used to process feces. There you can see salmonella getting black centered colonies. The last picture is Lowenstein Jensen medium. We generally used to have it in vials like this. We used to process TB specimens here. You can see buff colored colonies against a green color background. So the reason for having these in vials is, TB cultures should be kept incubated for more than 8 weeks. So to prevent evaporation of water and to keep the moisture, it is kept in tubes like this and maintained for longer duration. Right, so fungal cultures there we have special culture media, especially containing antibiotics. Usually takes a longer time to grow, up to 3 weeks. That's why there also we maintain them in vials. Isolated organisms are identified by morphology and some biochemical tests. So unlike now bacteria, I have to tell you, after we isolate them in artificial media like this, you see we have colonies. So these colonies are subjected to biochemical identification. Now, earlier we used to have conventional biochemicals. Then after that, we got semi-automated ones like API. Nowadays, we have fully automated machines called Phoenix, or we have Molitov, we have Vitex, but remember, not available in all hospitals. We have one at National Hospital Colombo, one at National Hospital Kandy, then one at Sri Javadhanapura and Apeksha Hospital. But other hospitals do not have these automated ones, so it takes time. So with automated ones, we can do it in 6 hours time, but after isolation, right? So after isolation, within 6 hours, we can come to a diagnosis. But with conventional methods, it takes another 24 hours because it needs incubation. So in contrast, when you want to identify fungal colonies, we always, most of the instances, we look for the morphology of the colony. So fungal colonies have typical morphologies which enables us to differentiate from another species of fungus, right? So it is very important. Basically, we go for the morphology of the colony. Now, when viral cultures are done, they are not routinely done in Sri Lanka. Now at MRI, viral isolation is done for acute flaccid paralytic disease because we have the polio reference lab in Sri Lanka, right? So viruses, require live cells for growth. So it needs cell cultures. So after going viruses in cell cultures, how do we diagnose them? We look for cytopathic effects of viruses. That means these changes happen in the cell line due to viruses are observed and examined. And also we can detect viral antigens. So say, for example, when we grow these polio viruses in cell lines, we expect these viral antigens to appear in the cell line. So we can have specific antibodies against the antigens and detect the antigen. And also we can do nucleic acid detection, that means PCR from this viral culture. So here I have given you some ways of identifying fungi. So when we get a skin scraping or thing like that, we have to pick up, you have to collect it to a clean paper, right? And fold it like a packet like this. Fold it to have a packet like this and send to the laboratory. Or you can have a container, sterile container like this. If you are putting it to a container, it should be a sterile container, right? Now these are the colony appearance. So you can go for morphology, and from the colony you can do microscopic examination. So there you can see different structures of the fungal colony. So you can see macroconidia like this, and tiny microconidia, and hyphae. So these are very characteristic features of a fungal colony. Right, so we have come to a very important topic again, antimicrobial susceptibility. So after identifying a patient having infectious diseases, say patient is having UTI, then you send a sample of urine to the laboratory, right? There we do culture. Then we isolate the colonies of bacteria from this artificial culture media. Then what we should do is we do ABST, antimicrobial susceptibility. Antimicrobial susceptibility is the main name of doing culture, right? So by doing that, we know what are the antibiotics that can be given to this patient to get him cured. So in vitro susceptibility is measured to guide the selection of the most appropriate antimicrobial agent that can be given to the patient, right? So antimicrobial susceptibility tests are performed by this diffusion method, broth and agar dilution method, minimum inhibitory concentration, that is epsilometer test, and also automated method, right? So out of these methods, what we have here is this diffusion method. What we do here basically in routine laboratory is this diffusion method. Sorry for having capital letters in the middle of a sentence. So this diffusion method is this one. So you can see we have done plating the organism on the plate. It's grown as a load. And then we apply before incubating it, as soon as the organism is loaned on the blood on the culture media, we apply the antibiotics and then we incubate. So following day, you see the organism grown as a loan, and around antibiotics, you see a zone of inhibition, right? So we measure the zone of inhibition ideally by using a vernier caliper, right? The diameter of the zone of inhibition is measured. And then it is checked with the standard table. Now we go according to the CLSI, that is called Clinical and Laboratory Standard Institute, which is originated in USA. There they give guidelines for this zone of inhibitions annually. So we have to now say, we already have identified this organism as E. coli. So we have to turn to the page of E. coli, or all Enterobacteria coming together. Then say this antibiotic is ampicillin. So we have to look for ampicillin zones. So if the zone is lying within the sensitive range, we can interpret as sensitive. So likewise, we interpret the zone of inhibitions like sensitive, intermediately resistant, and resistant. So you can see some antibiotics here around which you do not see a zone at all. So these antibiotics are resistant to this organism. So what we have to do is to report these to the clinician in a report. So the clinician accordingly will treat the patient with sensitive antibiotics, right? So this is the main basis of ABST. So I told you we can do detect the MIC also, right? So this is one method of detecting MIC. This method is called epsilometer test or E-test. So in this way, detecting MIC is easy. You can see this is clindamycin for Pseudomonas. Pseudomonas generally has a nice green pigment. You can see that here. You can see zone of inhibition appears as an ellipse. So when the two arms of the ellipse meet is the point of MIC. MIC has a better reflection of giving interpretation of sensitive, resistant, or intermediate compared to diffusion test. So MIC, we need to do MIC in such instances. We are compelled to do MIC. What are the instances? If the patient is having serious infections like endocarditis, meningitis, or infections at any sterile site, right? So now these strips are available in hospitals, but very, very, very expensive. So our strip is only for one antibiotic, and we cannot afford to do these for all patients. That is why we do MIC only in selected patients for selected infections. Invariably, we do it with infective endocarditis and with meningitis, right? Okay. Right. So, what are the non-culture techniques to detect components of the organisms? Now, these can be more rapid than culturing organisms. There we do antigen detection, as I told you earlier. So, detection of the pathogen-specific antigens using specific antiserum we can do, right? So, generally, we do this for CSF. Now, you would have heard about antigen detection tests in CSF. So, we have a small kit is available to detect 5 pathogens in CSF. So if a CSF, now it's again very expensive. Most of the instances we do not have it in these general hospitals, but available in private hospitals. So for Streptococcus pneumoniae, Neisseria meningitis, E. coli, Streptococcus agalactiae, and Listeria monocytogenes we do for 5 organisms. So it is very rapid. Before culture results appear, we can give a result to the clinician. But however, it doesn't have it doesn't have that much sensitivity. However, it is very specific, but even though it is negative, we have to do the culture. So toxin detection can be done for detection of pathogenic strains for causative agents, right? So it is done by direct detection of the toxin or its antigenic components of toxins, toxin-specific nucleic acid. So Clostridium difficile toxins, we do using an ELISA. E. coli toxins detection is done by DNA probes. So direct methods are done for toxin detection as well. And also identification of microbes by detecting their genetic material. So that is where we do polymerase chain reaction, right? So it is very highly sensitive and specific, but very costly. And it is rapid compared to culture, and quantification of pathogen load can be achieved by real-time PCR as well, right? Not only qualification, but quantification also can be done. So you can see, I'm sure you all know about the basis of polymerase chain reaction. First of all, you get denaturing of the organism. So, you know, you detect the DNA or RNA of the organism. If it is DNA, it is denatured using high temperature, right? Then this is denatured, and annealing of the probe to the target DNA is done, and then you can see elongation. So with elongation, you can produce more and more. Elongation is done. So when this cycle continues more and more, you can get more and more copies of the DNA of the targeted organism. Right. So we do serology to detect antibodies. So it's an indirect test, in fact. Not the antigen or the organism is detected, but the antibodies produced for the organism is detected here. In serology, acute infection is confirmed when you detect IgM. So you know IgM is produced first in an infection. So if you detect IgM, that means it is an acute infection. And also, if you see a rise of more than 4-fold rise of total or IgG antibodies within 10 days, then it is again an acute infection. And also, if you see seroconversion or appearing of antibodies, it is also an acute infection. So I have given you some ways of detecting antibodies. This is here VDRL. You can detect antibodies against a protein of syphilis, right? Hemagglutination inhibition test, precipitation test. Okay, so different methods can be used. ELISAs. Right. So, very important topic again, antimicrobial treatment. So, selective toxicity is achieved by exploiting differences in the structure and metabolism of microorganisms and host cells. So antimicrobials gives you a selective toxicity, right? So generally, organisms, microorganisms have different structures compared to eukaryotic organisms. So prokaryotic organisms are the bacteria. So we target against these bacteria using antimicrobials because they have selective toxicity. This is more likely to be achievable in microorganisms, right? Because, they have different structures, as I mentioned earlier. For example, mitochondria. Their mitochondria, ribosomes. They don't have mitochondria. They have ribosomes. So their ribosomes are different compared to ours. And again, they have cell walls. And so targeting these structures, antimicrobials act. At the other end of the spectrum, viruses are difficult to attack because they are obligate intracellular parasites. So a successful antiviral agent must be able to enter the host cell and inhibit and damage a virus-specific target, right? Some antibacterial agents are bactericidal, others are bacteriostatic. That means some antibiotics can kill bacteria, but some can only inhibit bacteria. So killing is bactericidal, inhibiting is bacteriostatic. Right. So antibiotic, mode of action of the antibiotic is different according to the classes of antibiotics, right? So some antibiotics inhibit the cell wall synthesis of bacteria. Some inhibit the cell membrane function. Some inhibit protein synthesis of bacteria. Some can inhibit the nucleic acid synthesis and some inhibit the metabolic pathways. Let's see. So here you can see cell wall inhibition. Let's see. Cell walls. This one. Second one from left. So beta-lactam, all beta-lactam antibiotics, vancomycin, daptomycin, telibacin, fosfomycin, they inhibit the bacterial cell wall, right? So inhibition of metabolic pathways, sulfonamides, trimethoprim, they inhibit the bacterial metabolic pathways. What are the antibiotics that inhibit the bacterial protein synthesis? Tetracycline, aminoglycosides, macrolides, clindamycin, chloramphenicol, linezolid. These are the examples to inhibit the bacterial bacterial protein synthesis, right? I'm going very fast. I don't think that you want to by heart these. That's why, right? But anyway, you will read these things from anywhere as well. But I thought that you have to have an idea about these things, right? All right. What are the antibiotics that inhibit the bacterial nucleic acid synthesis? Sorry. Fluoroquinolones, rifampicin, they inhibit the bacterial nucleic acid synthesis. Cell membrane function is inhibited by antibiotic like isoniazid, amphotericin B, polymyxins, these antibiotics. Right. So, however, let's see how bacteria develop resistance against antibiotic. So, when an antibiotic is developed, right? However, bacteria develops resistance against it. None of the antibiotic has escaped developing resistance against it. Okay, so this resistance can be inherent resistance or acquired resistance. So this inherent resistance is, some antibiotic act on different sides of bacteria. But bacteria, there's a target in bacteria. But what if a bacteria doesn't have the target? One example is this. Mycoplasma is a bacteria that doesn't have a proper cell wall. Okay? So, beta-lactam antibiotics act on bacterial cell wall. If mycoplasma doesn't have a cell wall, beta-lactam antibiotics will not be effective for mycoplasma. So we have to select some other antibiotic for treat infections caused by mycoplasma. That is inherent resistance. And bacteria can acquire resistance from other bacteria. Okay? So it can be due to chromosomal mutation or acquisition of foreign DNA. So this acquisition of foreign DNA happens due to 4 main mechanisms. One is conjugation, that means transfer of resistant plasmids. So then the transduction, that is transfer of genetic material by infection with bacteriophages. Bacteriophage is a virus infecting bacteria. So then bacteriophage can take resistant gene from bacteria to another. Transformation is acquiring DNA, resistant DNA from the environmental bacteria, other neighboring bacteria. Transposon is jumping genes, right? So, they have mobile genetic elements called plasmids. So these plasmids can jump from one bacteria to another. So how a person gets resistant bacteria? We have several bacteria, right, in our body. So say they may be sensitive bacteria or resistant bacteria. So I have given resistant bacteria in blue color. So say if we are exposed to antibiotics, right, these sensitive bacteria will get killed. Now say we are having pneumonia, right? So pneumonia can be caused due to sensitive bacteria as well as resistant bacteria. So when we get antibiotics, these sensitive bacteria get killed off, but resistant bacteria will remain. So this resistant, although this episode is over, the resistant bacteria can multiply and grow and continue to cause a disease due to the resistant bacteria. So this resistant bacteria can be transferred from us to another person as well, right? This is how bacterial resistance emerge. So these are the mechanisms of resistant bacterial resistance to antibiotics. Now I told you resistance can happen due to intrinsic resistance mechanisms and also due to so these resistant genes they acquire can present in many ways, right? So they can be inherently resistant or they can acquire resistant genes either by mutation or by acquiring of genes. So this resistance can be present in different ways. Either they can have efflux pumps, that means whatever entered into the bacterial cells can be pumped out. So fluoroquinolones are pumped out like this, aminoglycosides, tetracyclines, beta-lactams, macrolides, these can be pumped out through efflux pump, right? Some instances, they are not taken in. So penetration will not happen in such cells. So beta-lactams, tetracyclines, fluoroquinolones are not taken in due to closure of porins of the bacterial cell. And in some instances, you can see production of enzymes. That is the most important mechanism. So some bacteria can produce various enzymes. One is beta-lactam, right? And some bacteria produce enzymes against aminoglycosides as well. And macrolides, rifampicins. For all these, they can produce enzymes. You would have heard about beta-lactamase enzymes. So bacteria can produce more than hundreds of beta-lactamase enzymes for different types of beta-lactam antibiotics. And also they can modify their target, some bacteria act on cell wall, some act on nucleic acid. So fluoroquinolones, rifamycins, vancomycins, beta-lactams, macrolides, and even aminoglycosides are getting resistance against them, right? So these are the mechanism of them getting resistant. So now these are some important multi-drug resistant bacteria you come across, especially in hospitals. MRSA, these are resistant to all beta-lactam antibiotics. ESBL producing coliforms are resistant to up to third generation cephalosporins. And carbapenemase producing organisms are resistant to even carbapenem antibiotics. You know carbapenem antibiotics, meropenem, imipenem, ertapenem. That is the most widest spectrum of antibiotics we have. So that is the pathetic story. Right. So what are the measures to control antimicrobial resistance to us, in us? Antibiotic stewardship is the most important measure. So it is the effort to measure and improve how antibiotics are prescribed by clinicians and used by patients. So here we have to use antibiotics only when needed, only for corrected indications, not for all common colds and viral infections, right? Use correct antibiotic in correct concentration, in correct route. Appropriate duration should be taken, right? Use drugs in combination in specific situations like in treatment of TB. And very importantly, have to practice infection control measures to prevent spreading of resistant bacteria from one patient to another. And reduce the burden of infectious diseases. So you can improve sanitation on people and provide vaccination. So these people who are vaccinated will not get the infectious disease for which they are covered for. And appropriate use of antibiotics in animal husbandry, veterinary practice, and in agriculture. And also very importantly, regular surveillance and antibiotic use and antibiotic resistance. So have to do regular surveillances of antibiotic use and resistance. Then we can develop our own guidelines along with that. Right, a little bit of infection control, right? So very importantly, we have to have standard precautions. Standard precautions in infection control are guidelines to prevent healthcare-associated infections by all healthcare workers at all times when attending all patients. Very, very important. So there are 8 steps in standard precautions. Better to know all these 8 steps because it's very important. One is hand hygiene. Second one, personal protective equipment. Third one, respiratory etiquette. Fourth, sharp safety and safe injection practices. Fifth, cleaning, disinfection, and sterilization of instruments and equipment. Sixth, environmental cleaning. Seventh, linen management. And finally, waste disposal. So these are the 8 points. I will go through all these points soon. So first of all, hand hygiene, you know, effective hand washing is the one of the most important infection control practices, right? So hands are the important vehicles in transmission of infections, and transmission via hands can be minimized by appropriate hand hygiene measures. So you can do either hand washing with soap and water or use alcohol hand rub. But if the hands are visibly soiled, always you have to do hand washing, not alcohol hand rubbing, right? So, you know, there are 7 steps of hand washing, and even for hand rubbing, you have to apply all these, right? So I think healthcare personnel should practice all these 7 steps of hand washing. These guidelines are provided by WHO. So I will on my video for a while and give you an overview. So first of all, you have to wet your hands, and then you have to get the soap. So first of all, palm to palm, you have to rub your hands, right? Then back of the palm with the other hand. So you have to do this. Then this is the second step. Then palm to palm with interlaced fingers. You have to do third step. Then back of the fingers with opposing palms with interlocked fingers. Yeah, right? Then you have to pay special attention to thumbs like this and to the fingertips. And then we do washing the wrists as well, right? Then you can wash your hands and dry them as well. So you might wonder, why do we have guidelines to do hand washing? This is the reason. So in this picture, you can see red areas are frequently missing when we wash our hands, and purple areas are less frequently missing. And we wash basically the yellow areas. So it's very important to follow these guidelines. Okay. So all these steps of hand hygiene should be done in these 5 movements. So we call these as 5 movements of hand hygiene. So before contact of any patient, before any aseptic procedure, after body fluid exposure, after patient contact, and after contact of patient surroundings, we have to wash our hands with following all the procedures you saw in WHO guideline, right? So when we do hand hygiene audits in hospitals, we look for all these 5 movements of hand hygiene and whether they practice the steps, all the steps. Okay, right. So we have come to the next point of standard precautions, PPE. So we wear gowns and aprons to protect our skin and clothing. Now PPE, you know, basically to protect ourselves. So masks and respirators we wear. Then we wear goggles and face shields and gloves to protect our hands, right? Then we have respiratory etiquette. So if you have good practices in when you are having a respiratory tract infection, it limits transmission of respiratory pathogens spread by droplets or airborne routes. So we have to educate the healthcare workers, patients, and visitors to cover the mouth and nose with a tissue when coughing or sneezing, dispose the tissue to an infectious-based bin, and perform hand hygiene after contact with respiratory secretions, and wear masks and isolate the patients with respiratory symptoms, right? So this is the respiratory etiquettes. Then sharp safety and safe injection practices. You have to prevent sharp injuries, right? So all used syringes, needles, and broken glass should be collected into a sharp bin. This should be leak-proof and puncture-proof material. So when 3/4 of sharp bin is full, we have to dispose it. So safe injection practices. You have to take precautions when use needles, cannulas, and intravenous delivery systems. Aseptic techniques should be practiced to avoid contamination of sterile injection equipment. Not administer medication from the same syringe to multiple patients, even it is connected to cannula, right? Sorry. So my next point is cleaning, disinfection, and sterilization of instruments and equipment. You know, patient care equipment can transmit infections from patient to patient. They should be sterilized, disinfected, and cleaned accordingly. So when the equipment come in contact with patient's sterile body side, they should be sterilized. So surgical needles, scalpels should be sterilized. When the equipment come in contact with patient's body fluid, sterile, I mean, not in contact with blood or body fluids, but come in contact with mucous membranes, then you can do high-level disinfection. So endoscopes, you have to do high-level disinfection. Then equipment come in contact with patient's intact skin can be cleaned. Bed pans and things can be reused even with cleaning, right? They need not to have sterilization or disinfection. Right. So what about environmental cleaning? You know, environment play a vital role in transmitting of infections. They should be kept minimum minimum infectious by good design of buildings. So 90% of microorganisms present within the visible dirt. So we have to remove the dirt, and however, disinfectant shouldn't be used routinely, but you can mechanically remove the dirt. My 7th point is linen management. It is basically done in hospitals. So you know how to clean the soil linen. And for linen used for sterile purposes should be autoclaved. Then waste disposal. You know, hospital has 2 types of waste, hazardous waste and non-hazardous or general waste. So this hazardous waste are infectious waste, pathological waste, sharps, chemical waste, pharmaceutical waste, and radioactive waste. So in waste management, we have to do several steps. So first of all, waste minimization or separation, where you reduce the amount of clinical waste, right? So, you know, if you take a syringe and needle, what about the plastic cover of syringe and needle? It is not a infectious waste, isn't it? So you can put it in a separate bin. That means plastic waste bin. Then you can identify the waste according to a color code, and then waste handle. That means collection and transport should be done by trained people using specifically designated route to the storage area. So collected waste should be disposed within 48 hours of collection. So this is the color code. Now this is where we practice in Sri Lanka, right? So you can see the general waste in black color, infectious waste in yellow, sharps is in yellow and red stripes, red for glass, blue for paper, orange for plastic, and green for biodegradable waste. So first of all, you have to have an idea about disposal of sharps. So sharps should be disposed to a leak-proof container, as I told you earlier also. So sharps bin should be available at the point of use. When it is 3/4 full, it should be sealed and sent to incineration. Now the person who handles should wear heavy-duty rubber gloves. Then it should be incinerated. If any needle stick happens, you have to inform it. So when using sharp beams, never recap. Use needles. You can see you get needle stick injuries easily. Do not forcibly insert anything into the sharp beam. Do not overfill the sharp beam, and do not attempt to retrieve anything from the sharp beam. So a little bit about transmission-based precautions. So transmission-based precautions are used in addition to standard precautions for patients with known or suspected infection or colonization with transmissible or epidemiologically significant pathogens. So the type of transmission-based precautions assigned to a patient depends on the transmission route of the microorganism. So we have contact precautions, droplet precautions, and airborne precautions. You can learn more about these by visiting CDC sites. Okay, so from CDC itself, I got this poster. You can have contact precautions for some infections that are transmitted by hands, and the droplet precautions. So contact precautions are wearing gloves, washing hands, and droplet precautions, wearing, again, washing hands, wearing masks, right? Or goggles, face shields. Airborne precautions, again, cleaning hands, N95 masks to getting prevented from aerosols, and isolate the patients. Right. So specific preventive measures, active immunization or vaccination. It is one of the most effective public health tools. Vaccination exploits the ability of the immune system to develop immunological memory, right? And it aims to time the adaptive immune system to the antigens of a particular pathogen so that first infection induces secondary immune response. A vaccine can use live attenuated organisms, killed whole organisms, subcellular functions or antigens produced artificially by gene cloning or chemical synthesis. So live vaccines are more effective than other types, but carry the risk of reverting into wild type as well, right? So I have seen a person who has got polio due to polio vaccines. So you can see now we have removed serotype 2 from live polio vaccine and substituted with the killed vaccine, right? So that is the danger of live vaccines. So that is active immunization. Passive immunization is administration of immunoglobulins. Now, some instances we administer passive immunoglobulins for chickenpox. If the mother has got chickenpox, for the baby we can give, right? For hepatitis B, we can give immunoglobulins, and also in rabies, right? To prevent the person from a major exposure, we give immunoglobulins. So likewise, passive immunization we can do. And prophylactic antibiotics can be started in some instances. For example, if a patient is undergoing a major surgery, we can give prophylactic antibiotics. And if the surgery is contaminated surgery, then again we can give prophylaxis. Likewise, we give antibiotic prophylaxis. Right, that's all for the lecture. I'm happy to answer any questions if you have. Any questions, please?
Ma'am, can we have the lecture slides, ma'am?
Yeah, I will send a PDF of the lecture.
Thank you. Thank you, ma'am.
Okay. Okay, in the absence of questions, shall we wind up? All the best to all of you.
Thank you, madam. Thank you, Nizri.